many attendees who have joined both on Cisco Webex and YouTube platform. Welcome to you all. Good morning. KST has been organizing Nobel Lecture Series for the last 3 years. It was started on the suggestion of former Vice Chancellor of Bangalore University and former Director of NAC, Dr. H.K. Ranganath, who was a former member of KSTA. And it was overwhelmingly supported by the previous KSTA committee, chaired by Padma Shri Professor S. Ayyappan, former Director General, ICR, Secretary, Department of Agriculture, Research, Education, Government of India. Some of you may be aware of KSTA. KSTA was started in the year 2005 under the chairmanship of renowned space scientist and former chairman of ISRO, Professor U.R. Rao. From 2005 to 2017, we worked under his guidance. Then another great space scientist, the architect of Bengaluru Deep Space Antenna and former director of IISc U.R. Rao Satellite Center, Dr. S.K. Shivkumar, Padma Shri Awardee, was our chairman for 1.5 years. After that, Professor S. Ayyappan, former DG ICR, was our chairman from 2020 to 2023. Under his tenure, this particular Nobel Lecture Series was started. As we know, Nobel Prize is a prestigious international award established by the will of Alfred Nobel, a Swedish inventor and philanthropist, in 1895. It recognizes outstanding achievements in 6 categories like peace, literature, chemistry, physics, medicine, and economic sciences. Each award consists of a medal, citation, and cash prize. The Nobel Prizes are awarded annually, with the Peace Prize presented in Oslo, Norway, and the others in Stockholm, Sweden. The recipients are selected through rigorous process by various committees and institutions. In fact, Nobel Prize in Chemistry has been awarded 116 times to 197 Nobel laureates between 1901 to 2024. Among them, Frederick Sanger and Barry Sharpless have both been awarded Nobel Prize in Chemistry twice. And sometimes the prizes, as I told, are shared by multiple laureates. And this year, Nobel Prize has been shared by 3 eminent scientists: Dr. David Baker, who works in University of Washington, Seattle. He is a professor there, and he got Nobel Prize for computational protein design. And the second half of the prize goes to Demis Hasnabis and John Jumper. Both of them work at Google DeepMind in London for their work on protein structure prediction. And now Madam will be speaking on protein structure today. And before beginning her talk, let me give a brief overview of Madam's achievement. Dr. Nagasuma Chandra Madam is a professor at the Department of Biochemistry and additionally affiliated with Bioengineering and Mathematical Biology Initiatives at the Indian Institute of Science Bangalore. She has contributed immensely to the development of bioinformatics and established the area of molecular systems biology in the IISc. Her research is interdisciplinary and encompasses algorithm development for large-scale analysis of protein structures, structure prediction at a proteome scale, structure-based function prediction, and their integration into cellular level understanding of biological systems. She is an elected fellow of the Indian Academy of Sciences and the Indian National Academy. Dr. Nagasuma Chandra Madam obtained her PhD from University of Bristol, UK, and subsequently moved to the Indian Institute of Science Bangalore. She has published more than 175 scientific papers in national and international journals of repute. She has mentored about 25 PhD students and several postdoctoral fellows, and she has served as an associate editor for current research in structural biology, and also she is a guest editor of current opinion in structure biology. And with this brief background, And in spite of Madam's very busy schedule, she has been very kind enough to accept our invitation to deliver the talk. And she will be speaking on protein structure, as I told. With this brief introduction, I request Madam to take over and deliver her talk. Over to you, Madam.
Thank you very much, and it's a great pleasure to be speaking in this forum. I thank KSTA and the organizers. Are my slides visible?
Yes, Madam, visible.
I'll put it in slideshow mode.
No, Madam, slides are not yet visible.
How about now? Now can you see my slides?
No, ma'am, not yet, ma'am.
Oh, I see. Okay.
Madam, slides are not visible.
Visible? Okay.
No, not visible.
Oh, okay. Let me just try again. How about now?
Yes, ma'am. Now.
Yes, madam. Yes. Thank you.
Okay. Wonderful, wonderful. Thank you so much. So as as the chair kindly introduced, the Nobel Prize in Chemistry has gone to 3 remarkable scientists. So the citation reads like this, they cracked the code for proteins' amazing structures. So it's all about proteins, proteins are life's ingenious chemical tools. And the 3 scientists are David Baker, Demis Hassabis, and John Jumper. They have succeeded with the almost impossible feat of building entirely new kinds of proteins. This is David Baker. And Hassabis and Jumper have developed tools, AI-based models to solve a very long-standing problem of predicting proteins' complex structures. So, and of course, the discoveries hold enormous potential. So let me just take you through some of these things. Yeah. Now, David Baker was born in Seattle. So I'll just get the view. Okay, fine. And in fact, trained in Harvard as an undergraduate in biological sciences, and then in biochemistry at Berkeley. He obtained his PhD. In fact, many of us have had an opportunity to meet Baker recently, and John Jumper as well, even more recently in the last few years at IISc when they visited separately. In fact, my former PhD student was hired by Baker when he visited us, and as a postdoctoral fellow. One of our undergraduate students as well, after he's graduated, is currently doing a PhD in Baker's lab. So we've had this exchange, which we feel very proud of, really. Okay. Demis Hassabis also has had a very different type of training. He was a genius as a kid. He designed video games even before going to college. He was trained in computer science. And then he returned to graduate school after an entrepreneurship stint to do a PhD in neuroscience, and then founded DeepMind. And as they say, the rest is history, really. And John Jumper, now he was trained as a physicist and physics and mathematics was his initial training, specialized in condensed matter physics, and he obtained a PhD in theoretical chemistry, and he joined DeepMind, and they collaborated on this wonderful work that has won the Nobel Prize. Right? Now I understand that I'm talking to a diverse set of audience in order to pass on the excitement of the achievements of these Nobel laureates, to appreciate the magnitude of their accomplishments. I feel it's essential to understand a little bit about the background, about protein structure, and how the field itself has developed over the years, right? And the state when Baker, Hassabis, and Jumper made their pivotal contributions, and what were the breakthroughs and what were the major enablers. That's the excitement that I want to pass on today in this lecture. I hope my slides are visible. They are moving.
Yes,
you can see the next slide. All right. Okay. Okay. Now, these are molecular machines of life. As you all know, proteins are the workhorses in biological cells. They come in different shapes and sizes. So all these drawings that you see here are different shapes, three-dimensional shapes of protein molecules. They come in a variety of shapes, sizes, anything that you imagine, any wonderful pattern that you imagine is already there in nature, in the form of a protein molecule. Very exquisite designs, they will make the exquisite rangolis that we see here look pale in front of the designs that you see in nature. Very intricate patterns, if you will. So what I'm referring to obviously are the three-dimensional structures of these protein molecules. Just a glimpse of them is shown in this slide here. But then you may ask, why do we care for protein structures? Why do we care for structures, right? They definitely make pretty pictures, but is that all? Of course not. So the structure is now important for the function. What proteins are the workhorses that carry out a range of functions. In fact, anything in the body is, anything in a biological cell is due to the proteins. So they digest the food, they take care of the immune responses that we see minute by minute, hour by hour, day to day that we see. We encounter a variety of pathogens and respond to it and stay healthy. And that's the work of proteins. Their hormones, they regulate, for example, how much insulin should be produced when we eat and how much glucose should be put back into the system when we haven't been fed. All these kind of things are carried out by protein molecules. Okay. So variety. In fact, it is not an exaggeration to say that the highest level of anatomical description you can have of any living process in any living biological system is at the level of three-dimensional structures, as you see here. Okay, this is just an example. An organism made of organs, of tissues, cells, organelles, supramolecular assemblies. At the end of the day, the detail that you're looking for is at the level of molecules, macromolecules, which are proteins and nucleic acids, which proteins are the workhorses which take on carbohydrates, nucleic acids, lipids along with them in order to perform various functions in the system. Okay. And what we are seeing here is a three-dimensional structure of a protein called, as an example, I put out something that's known to everybody called cyclooxygenase, but the drug will be better known to people, which is brufen. We all take brufen when we get pain. What you see in a little blue color, cyan color spheres out here is the drug called ibuprofen, right, which is in the tablet brufen. And this drug molecule binds to this protein called cyclooxygenase and inhibits pain and inhibits prostaglandin synthesis and thereby inhibits pain. That's how we get relief from pain from them. At the heart of this are amino acid residues from proteins which interact with this drug molecule or whatever natural function that the thing has to do. So the highest level of anatomical description of any living process, whether pain, whether digestion, whether thinking, whether, as I'm speaking, as you're listening, what are the kinds of protein interactions that are going on is all described best at the level of three-dimensional structures of proteins. Okay. Now, proteins are synthesized as linear polypeptides. Again, this is well known to everybody. Proteins are polymers of amino acids. So when they're synthesized in the cell, they are linear polymers formed. Amino A1, A2 stand for amino acid, and peptide bonds connect them. They're like a linear chain, like a chain of beads. But in reality, they don't exist as a chain of beads. They quickly assemble in a very, very specific manner to become what we call as the tertiary structure, and then in some cases, the quaternary structure of protein molecules. So to perform their function, it is this either the tertiary or the quaternary, depending on the protein, that is necessary for this structure to form in order for the protein to function. This is an example of hemoglobin that you see here. We all know what hemoglobin does. It carries oxygen to every part of the body. In order for hemoglobin molecule to bind oxygen, this linear polypeptide chain must fold into this very specific pattern, tertiary structure, and in this case, multiple subunits come together forming a quaternary structure. That's what it is. And given the importance of structural biology, there has been a lot of attention to solving three-dimensional structures of proteins. It's a very hard task, and that's reflected in the number of Nobel Prizes that have gone for the discovery of various protein structures and discovery of biological insights based on these protein structures. Starting from, the first crystal structure that was solved by Max Perutz and John Kendrew. In fact, I was fortunate to be a student of Max Perutz's student, okay, in the sense an academic grandparent, and the number of Nobel Prizes. What I have put down here is what is relevant for today's talk, some of the key milestones that ought to be remembered each time we talk about protein structures. I will come to some of them as we go along, especially Christian Anfinsen and Martin Karplus. We will touch upon that as we go along. But there have been notable discoveries and many Nobel Prizes for discovery of protein structures. I think my cursor is not working. I'll go slowly about this. Okay. So some of the other milestones of protein structure, again, we've had a great connection with Dorothy Hodgkin, who won a Nobel Prize for solving the structure not of a protein, but of a large molecule, which is vitamin B12, using X-ray crystallography. And we've had my postdoc advisor, Professor Vijayan, was a student of Dorothy Hodgkin. So we've had that legacy at IISc. Many of us have worked with people connected to these great scientists. And there have been many of other notable scientists, Hendrickson, Helen Berman. Many of these things have advanced the field. What you're seeing in the slide here is how crystallography has evolved over a period of time. And that was the main technique or tool that was available for determining three-dimensional structures right from 1965 to 2005 and then to the current day. A lot of advances have happened, starting from say myoglobin to lysozyme to a virus structure, and I'm sure you'll hear much more in Professor Murthy's talk tomorrow, and a variety of other molecules that you see out here, to the ribosome, for which Professor Venki Ramakrishnan won a Nobel Prize. So one of the main things that has helped the field, other than development of crystallography as a technique, is also making all this data accessible to the community at large. So a protein data bank was a database where everything was beautifully archived, and to date it continues to be the main source of information for anybody in the field, whether to learn protein structures or whether to apply them for a variety of purposes, as we will see. And Helen Berman is one person who's really contributed quite strongly to this endeavor. Okay. Now, understanding how proteins work. So I've taken an example again of a T cell. T cell is a T lymphocyte, our immune cells, basically, which is there in our bloodstream. This is the picture of a T cell. Now, what T cells do is to attack pathogens, attack viruses. And when we say cells attack viruses, it's not the cell by itself. It is the proteins that are there in the cells that do this job. Right? And what you see in the panel below here is a T cell receptor, which on the T cells, which are being recruited by what we call as antigen presenting cells. Now, we're all familiar with the COVID pandemic that hit us recently. Now, the COVID virus will have some specific stretches of peptides, which are recognized as antigens or as a foreign substance by our immune molecules. And in this case, what we call as the HLA or the MHC in orange. I don't know if my cursor is visible. Yeah, this orange one is the MHC molecule. This blue one here is the TCR, or the T cell receptor that comes from the T cell. This is coming from another cell which binds this COVID virus antigen, which is shown in green out here. And I'll show you a little video in a minute. And this recognition is leading to a range of biochemical effects, as you'll see in a picture below. I won't go through the details, but it ends up finally in the T cell. If we are lucky, if we are successful in launching an immune attack, it will go and kill the infected cell, the cell which infected with the virus, and thereby kills the infection. So what's important for this process is a form. When we say form, it is the three-dimensional structure of the protein. It is the dynamics of that. It's not a static molecule. it is dynamics. It changes shape slightly and then adapts itself to the requirement, of course, based on how it has been programmed to do so through evolution. And it's a functional cycle that you will see. Yeah, there's a little video. For some reason, my keyboard is not responding. Bear with me. Yeah, I think this is better. I'll just come out of this mode for a minute and show you the video. Yeah, something is wrong with the video is not working when I open it, but never mind. What I wanted to show you was how this process of HLA molecule on the right-hand side binds this antigen and how the T cell receptor gets recruited, or these are two different proteins we are talking about here, binding an antigen and then performing a certain function. What I wanted to point out here is, on the left-hand side, what you are seeing is basically the three-dimensional shape in a certain way of visualizing this protein structure. And it's exactly the same molecule that you see here, but visualized in the form of the skeleton, or what we call as, it's a cartoon diagram representing the skeleton of this protein molecule for simplicity, because it becomes very complex to understand what the protein is doing when you look at it as an agglomeration of various spheres, which are atoms coming together. Whereas in this representation, which is what you will see later on also, it's the skeleton of the traversal of the polypeptide chain, which we refer to as C-alpha trace. And that helps us understand what it is. Now, just to touch upon the importance of protein structures and to what level of detail that we want to understand, it's the same HLA molecule here. Each of us carry in fact, 6 different alleles, slightly different variations we will have among us, amounting to thousands of different alleles of these HLA molecules. And each of us can respond differently to different antigens based on the slight difference that you see. What I am showing here is only a portion of this protein molecule that is directly responsible for function, which is shown in red color out here. What is in yellow is an antigen that can come from a virus or can come from a cancer cell or whatever. So it's the three-dimensional structures at this binding site, which is responsible for very specific function. Who mounts a stronger response, who mounts a lesser response, and so on, is coming from this particular small change in that. Talking about it differently, now glucose, we all know what glucose is. Glucose is recognized by, I haven't shown the protein out there, let us say glucokinase, hexokinase. Glucose binds to that and serves as fuel and produces ATP for us. Whereas a closely related monosaccharide, mannose or galactose, will not be recognized by the same protein in the same way. And therefore, it goes to the extent of the implications are so deep that glucose is life-sustaining and is food for us or for bacteria, whatever, whereas mannose or galactose are not. They have different functions, but they are not the major sources of carbon sources and major sources of energy, right? So now, after talking to you about the importance of protein structures, why is structure prediction so hard, right? In fact, experimentally, structures are determined through crystallography, some through NMR, and now more recently through cryo-electron microscopy. I will again touch upon that in a minute. But why is prediction so hard? Now, the complexity in structure prediction is very high, right? So what I'm showing here is a small portion of a polypeptide chain, residue 1. When you say residue, it is amino acids, amino acid 1, amino acid 2, 3, and 4 here, and again 4. A small change in this position, nothing has changed. Bond lengths haven't changed, bond angles haven't changed, but the orientation of one residue with respect to the other has changed here, which is based on torsion angles or dihedral angles. Just the change in this dihedral angle that you're seeing here it is giving a different shape in this portion. And the effect of that is what you are seeing here. So one portion of the protein will be the same, but the rest of the portion is different because it's gone off in a different orientation altogether here. So it can have, these torsion angles can have huge changes right? And now we understand, let's consider an example of a 100 amino acid chain. And each residue is a technical term here. It's an amino acid is what you can think of. Each amino acid can exist in one of the three broad, what we call as secondary structure states, alpha or beta or X for anything else out here, a random coil. And if we consider three possibilities for every amino acid, we will still get 3 to the power 100 conformations for a 100 amino acid polypeptide. It's a huge number. This roughly translates to something like 5.1 into 10 to the power 47 different shapes, approximated to 10 to the power 47 shapes. And these conformations, these torsion angles on of the backbone of the protein, they are referred to as phi and psi, and they are in the famous Ramachandran map after the discovery of G.N. Ramachandran, where phi is on one axis, psi is on another axis, and only some portions of this polypeptide of this phi psi plot are occupied by natural proteins because they are the ones that are stable. So we understand a lot about that. In fact, this type of a calculation not only shows you why structure prediction is hard, but has led people to wonder, how do proteins fold in nature, right? If there are 10 to the power 47 different conformations a protein has to actually explore, because it's synthesized chemically in the cell as a linear polypeptide chain, but it needs to go back into a three-dimensional shape very quickly, which happens in fraction of a second. How is that even possible? In fact, that's the famous Levinthal's paradox, because the amount of time that may be required to explore this space is more than the number of atoms in the universe, it's often said. So how does it even do that, right? But then the paradox is resolved by understanding that it happens in a hierarchical process, more like a funnel from an unfolded to a folded state and so on. So there's an energy landscape which drives protein folding in nature. And some of it is very useful for us to understand, to apply it in prediction. Okay, so now we have actually, I'll just come to this in a second. My slides are okay. So now linear polypeptide chains we refer to as the primary structure or the sequence of the protein molecule. When we say sequence, it is the order in which amino acids are connected covalently with each other to form a 1D structure, or what we call as primary structure, or very commonly referred to as the sequence. There were very elegant experiments done by Christian Anfinsen, who won a Nobel Prize in 1972, by taking a protein called ribonuclease, denaturing it very carefully, and then showing that the function of the ribonuclease was lost. But then this was all in a test tube. This was not in a cell, in a just a test tube with only the protein and the buffer molecule, buffer solution, and so on. But they renatured the protein very, again, carefully, and they showed that the activity was regained 100%. It might, at the outset, you may think, what is the big deal? It was denatured, and therefore the word denature itself means that protein function is lost, and it's renatured, it has come back. But it is actually a very profound theory that came out of this theory, and which was nicely proven later in many cases. So this is in a test tube, not in a cell. The only information that the protein had to go back into it properly native state is what we refer to it as, properly folded state, is the primary structure or the sequence of the protein. First of all, it showed that if you disrupt this structure, function is lost, which means structure determines function, is a very beautiful conclusion that came out of this study. And the fact that it is folding back into its native state means that the information, the instruction for folding it back must be available in whatever the test tube had, which was only the protein molecule. And the protein molecule is defined by its primary structure, and that was the sequence. So sequence determines structure was very clear, clearly determined from this exercise. In fact, that is what laid the foundation for bioinformatics as a discipline, came up later. So that was in fact, Sanger, I must mention Sanger, who won a Nobel Prize, once for determining the structure of insulin, but later for determining the sequence of this protein in the form of nucleic acids, which later converted into protein. So the whole idea of sequencing as a DNA, as a sequencer, was started through discoveries of people like Sanger. So you can actually read out what is there in a nucleic acid chain, a gene which codes for a protein. You can read out the gene sequence very nicely through automated machines today, which are available. In fact, the latest machine that I do have in my lab it looks like a small pen. It's from Oxford Nanopore, a tiny sequencer which you can hold in your palm and then do all the chemistry that is required to determine the sequence of a gene molecule or an entire genome of an organism. So from moving from a single gene, we moved on to the era of genomics now. And the Human Genome Project is one of the few which has been completed ahead of schedule and with lesser money, that I'm told, to read out the entire genetic material of the human genome and many, many other organisms' genomes, many bacteria, many other eukaryotes, plants, all of them have been sequenced now. Okay. So now we can go from the genome to each gene computationally. You can identify that using, and from gene to protein sequences using the standard genetic code. One can computationally derive that very easily, which leads us to a large number of protein sequences. Again, like in the protein data bank for three-dimensional structures, we have very beautiful databases accessible to anybody. You can access them through your mobile phone if you like, which are in databases, launched at NCBI in the United States or in Europe, EBI in UK, or the Swiss Prot in Switzerland, and so on. So you'll see the number of sequences that are known run into millions out here, right? Millions and millions of proteins are known out there. So this brings us to the situation of having so many protein sequences, which is represented as a sphere here. But the number of three-dimensional structures that are actually solved experimentally, either with X-rays or NMR and electron microscopy, are a tiny minuscule of this entire thing, like 0.1, 0.05% or something of that order. This light green pie that you see in the first pie chart here is what we actually know. This is the sequence structure gap that we are seeing. There's so many sequences, but very few protein structures. We'll come to the other one in a moment. But then we know that sequence determines structure, okay? Which means, as a corollary, that similar sequences will have similar structures, right? So there, of course, a huge number of technical challenges, how to find similarities in sequences. What does similarity even mean? How to find similarities in structures for us to answer the question completely, okay? And fold versus atomic structure. The skeleton that I referred to is referred to as a fold, whereas the fine structure with all atoms filled in is referred to as the atomic structure. Again, how similar is similar? How do we quantify it? And so on. There are many challenges that have been solved along the way, including understanding whether it is a result of divergent evolution or convergent evolution, and how do we make use of this information for understanding protein structure. So one of the names that I must mention in terms of utilizing this principle very nicely, similar sequence determines structure, or similar sequences have similar structures, is, should be credited to Andre Sali and Tom Blundell, who was his mentor. Andre Sali developed the Modbase, or a modeler software, which represents this idea of homology modeling. We say that if two sequences are similar, we refer to them as homologous. They're evolved from a common ancestry and so on. And therefore, their structures, at least at the level of the fold, should be similar, is what we say. And this is a computational pipeline developed by Andre Sali, which addresses the sequence structure gap to some extent here. So if you do homology modeling of all these 600 million sequences, you can fill up much more of this pie chart out here with all these orange color sectors, because there are examples which are fairly easy to model based on simple principle of similar sequences or similar structures. The procedure itself has number of computational steps and number of innovations, number of pipelines by many people, including by David Baker, which is called Rosetta, that came a little later, although I will touch upon how they used it for a slightly different purpose later on. Okay. And there are many physical principles that are considered for protein structure prediction. I won't go through, in the interest of time, all of them. But of course, the forces that hold a protein together, hydrophobic effect, and many non-covalent interactions like hydrogen bonds and van der Waals interactions and so on. And free energy minima, the key principle is to have the least amount of free energy and to get a stable structure, and considering solvation, entropy, and evolutionary constraints and so on. So many methods were, because of the sequence structure gap was so wide, structure prediction has been of interest, and many methods have been developed for structure prediction, including homology modeling that I just touched upon. There are others as well, but now, in fact, those use the principles of what we call as multiple sequence alignment. You take a protein and look at it from multiple species to see which amino acids are conserved. What you see on top here are amino acids, a portion of a protein molecule. And each row represents the same protein from a different species. One can be from a bacteria, Mycobacterium tuberculosis, other from E. coli, other one may be from human even, because there are many common proteins out there. And the letters represent amino acids. You know, there are 26 amino acids, and there are 26 letters in the English alphabet, beg your pardon. There are 20 amino acid letters. So we use 20 of the 26 letters out here to represent polypeptide chains. D, for example, stands for aspartic acid. When you see D out here, that means it's a conserved aspartic acid in that position in all species and proteins from various species of this protein. And this has a lot of information that one can make use of quite quantitatively through various matrices, scoring functions, and computational algorithms in order to get insights about what is important for protein function. Okay. So, and in fact, looking at these kind of computational methods, the beauty of that is you can scale up very easily from one protein to many and find general patterns. This is a nucleic acid, but you can do the same in proteins and look for generalizations in biology. And this is where bioinformatics has become established as a discipline. Our own little contribution to homology modeling, because it had become so common. Many people have been using it for a while now. This was more than 10, 12 years ago, 15 years ago. And able to model three-dimensional structures of majority of about 70% of the genome in the sense of proteome of Mycobacterium tuberculosis. And that is what you see out here. These are all the structural models out there, and you get details of them and try and understand what their function and so on and so forth. So that's a little contribution that we had. And what does that lead to? It leads to understanding patterns which are predictive of protein function. What amino acids are important for the fold, portion of the fold, what do they recognize, and what is their function, and so on and so forth. But then a huge leap actually took place, which is, I'll touch upon the work of John Jumper and Demis Hassabis first, because they used AI, right? Physics-based modeling and bioinformatics in the traditional sense used all of these kind of methods. They use computational methods. But to scale up from what we were doing at that time to what is currently there is because of these huge advances with huge benefit with AI. Now, what is AI, right? Artificial intelligence, you refer to that as any technique or system that allows computers to mimic human behavior. Okay, but then the way to make the best use of it is through machine learning, which takes in large quantities of data and then finds patterns and then solves the question that you have in mind, basically. And one of the ways of doing that is through neural networks. So in any deep learning is a very specific branch of machine learning. In any machine learning exercise now, you have training data like known knowns. You have data where you have known labels for them. Then, of course, a lot of data analysis has to happen, processing, normalization. You must make the data amenable for comparisons and so on. And remove the noise, keep only what is important, so dimension reduction and so on. And you need to learn, you need to train the system from that. The system learns from it There are many ways of learning, supervised, unsupervised, and so on and so forth, and estimate the error, which is a very important part of any machine learning exercise, to become now a predictive model. So once learning has happened, if there is an unknown data with unknown label or new data, you plug it into the system, it predicts and tells you what it is, right? Now, AI has now, it transcends all disciplines, all spheres of life. Whether you're shopping for something in Amazon, it gives you, you may like to see this and so on, that kind of a suggestion. Or whether you're doing deep science, such as understanding protein structures or whatever, it tells you what is the best way forward. So it gives you a solution for it, right? And what has helped in protein structure prediction by Google's DeepMind is this neural network, where they modeled it as protein sequences were taken as an input, and then they modeled it in a form where patterns could be identified. Basically, they applied deep neural networks to capture the complex relationships between amino acid sequences and protein conformations. So what you see here, there are layers of them. These layers are key components of a neural network. Layers are also referred to as neurons because they are inspired by how our brain works. That's why they're called neural networks. These are artificial neurons in a neural network. They consist of, they're interconnected, as you will see. And each neuron is a processor. It processes information from the previous layer and passes it on to the next, as you will see here, and so on. And for protein prediction, structure prediction, the network learns sequence-specific features and how they contribute to structural motifs and so on. So the input will be a protein sequence, because we know that the sequence world is so vast now. We have sequences of almost, of every known protein, for example, or every conceivable protein from an organism. The idea is now to get structural information from that and then get biological insights from. So that's the input, and the output we want for this exercise is a three-dimensional structure. So there is, you will see there are thick lines and there are thin lines. These can represent the weights, how much weightage to be given for what learning is happening, what information is there, and so on. In fact, our brains also learn in the same way. We leave out trivial pieces of information which we think is not useful to remember something. We will only remember the key points and so on. Right? So that's the thing out here. And AlphaFold is the name of the tool. AlphaFold itself has now moved on from version 1 to 2, and the 3 is the latest one that is going on, which was Google acquired this DeepMind company, and therefore it's become Google DeepMind. Since their breakthrough, AlphaFold has been used by more than 2 million people from 190 countries, which is almost all the world, really. So what you see here is a picture of RNA polymerase, a protein called RNA polymerase, okay, where the predicted one is in blue from AlphaFold and the actual structure is in green. Okay, the actual crystal structure is in green, and they're superimposable so well that you can't tell the difference between blue and green. Only the colors that you see is because of the depth rendered onto a plane of the screen out here. So if you rotate it, you will see that the green and blue overlap very, very well. And what AlphaFold 2 has led to is increasing the structural coverage. The sequence structure gap has been filled up almost entirely. The pie chart is now not empty at all in many, many species. Right? The AlphaFold database, if you look at it, is almost completely full. The structural coverage is very high. Like human genome codes for about 20,000 proteins, and you will see the bars stopping somewhere there, here, right? Whereas plants have larger number of proteins they code for, and therefore you will see many more like that. But bacteria, the coverage of the proteome now is almost complete, right? So how does AlphaFold work, right? AlphaFold now considers evolutionary, physical, and geometric constraints, right, in order to predict the three-dimensional coordinates. And the accuracy is very high. Accuracy is so high, I'll just come to that. So it uses a novel neural network architecture which jointly embeds these kind of information, evolutionary in the form of multiple sequence alignment and residue pair information and so on. So what it does is, it's in fact a significant leap in protein structure prediction. So it has a number of modules called the EVO former module, which handles all this evolutionary information in terms of, is this protein conserved across different species? And if so, we know that the function is conserved. And if so, what are the sequences? What are the amino acid residues that are conserved? And how can I make use of that information in training the network, right? And a reference database is made to capture all of these things. It has a deep neural network module for learning this kind of information, and a structure prediction module, which makes use of the physical principles that I talked about in terms of forces and all of that, and the geometric constraints that are there into structure prediction, and it refines it until it gets a very good model. Okay. What you see as a picture out here is something called a 2D contact map, which we have been looking at for a long time. If it is a 50, this is a small protein, a 58 amino acid protein, you write them on one axis and write the same on the other axis, and which amino acid residue is in the spatial vicinity of which other is what this picture is actually capturing. In other words, it's making a 3D map into a 2D map out here and telling you what the adjacency of amino acid residues are in the protein structure. Now, this information can be overlaid with which two amino acid residues co-evolve if you look at a multiple sequence alignment and so on. And that is what John Jumper and colleagues and Hassabis have put into this particular AlphaFold algorithm. They identify co-evolutionary signals, multiple sequence analysis, they call it as pairwise residue analysis. So if in a, amino acids keep changing as evolution takes place, even in a bacteria, if we go on culturing the bacteria and make newer and newer generations of that, some amino acid residues change, but those don't have any contribution to the function, in the sense those don't alter the function. Any alterations to any essential functions that are there, evolution sees to it that such sequences are not tolerated, and therefore they're eliminated from our list. Like what we see are only stable sequences, only where function is retained. So when two amino acid residues are mutated codependently in different homologs, the residues will be in close contact. So they make use of this information by saying that if two positions are changing simultaneously and retaining function, the two should be in contact and so on. I won't go into the details of it in the interest of time, okay? But that's the kind of things that they actually put in place here, which has gone into a deep attention-based neural network. So AlphaFold architecture actually makes use of what we call as a transformer model, right? A transformer model now is nothing but paying attention to the key things, and it's a neural network model, but it's called as an attention mechanism, and therefore they call it as a transformer, where the mechanism allows the network now to focus on the key bits, the specific parts of the input sequence that are important and so on, right? In fact, that we do that all the time. When we see somebody, we remember only certain things and not all. We don't remember, for example, the color of their clothes that they wear, but we will remember where we met the person, right? And in what context we met the person and what they do, what is their job and so on. We will remember those things. We pay more attention to certain things than other things. That is what this is actually learning automatically and then doing. Multiple sequence alignment and a huge database and key weights, the EVO former and the structure module, like I said, and it iterates until it is happy with the structure it has produced using this neural network architecture. So I will skip the rest of it since, It also has something called a teacher-student model, again, part of the neural network training, where a teacher model now is a very accurate, large training model. Okay, it takes time. Whereas a student model aims to mimic, it comes hierarchically. It aims to mimic a teacher model, but with lesser data and but much more efficient. efficient because it's using less data and coming up with a solution. It may be as good as the teacher model, but that's good enough, for us to do the job. So it uses hierarchical modeling of this kind. And it also has a way of scoring for where it is correct, where the structure prediction is correct, where the structure prediction is wrong, in terms of an estimation of confidence. We call it as PLDDT here, predicted local distance difference test. But and you will see different color coding out here. If the model is highly confident, if the score is greater than 90, it's deep blue. Where it is very poor, it is red out here. So what we do, there are regions which are poor in some protein molecules, depending on what it has learned from, depending on what the protein data bank itself has. Okay, so currently there is still scope for improvement. Currently, many models have blue, light blue to dark blue, and so on. But there may be portions which are like this looping all over the somewhat like spaghetti floating around, which are not confident parts of the molecule. We don't consider them for analysis or for anything currently, but there is a good chance that it will be improved as we go along. But it is this blue part that is very precious for us out here. Okay, and there's a way to predict the error. How is error predicted? Again, you will see the expected error. The more green it is, the more confident the error is less in the model. And the way to estimate error is by comparing known structures with the predictions of known structures and so on. In fact, people did an experiment called CASP. CASP is now a critical assessment of structural prediction, and it has been going on for nearly 28 plus years, right? Since 1994 or 1995, it's been going on for a long time. John Moult is a key figure who started this process. Again, this is a video, I think might play. Yeah, it shows you how the AlphaFold prediction is actually getting the model right, and then comparing the way the experiment is done. Sorry, I should explain that. Is there are people who are experimentally determining protein structures, but that information is kept hidden, and only the sequence is given to modelers, asking them to predict the structure. Now, the predicted structure is matched with the known structure kept hidden. So let's go back to this. Yeah, I hope my slides are still visible. And then compared with, the predicted structure is compared with the known structure. Another video, but I will skip showing that. So CASP experiments have been really beautiful and helping in understanding where we stand in the field. Now, the 14th cycle of CASP has already happened. AlphaFold has performed very well in CASP 13, where we initially participated, and CASP 14, it was a groundbreaking kind of advance that they made out there. So by looking at how well the prediction is happening without the knowledge of the experimentally determined structure, you're able to assess what is the value of prediction, right? And if you can do it for one protein, you can do it for all proteins. That is what AlphaFold has actually demonstrated. Now, in this bar out here, AlphaFold is here, and all other prediction methods are on the right-hand side. The lower the y-axis here, lower the value on the y-axis, the more confidence the model, the less the error. And AlphaFold is less than 1 angstrom. 1 angstrom is, 10 to the power minus 10 centimeters. So small it is. It's like a bond distance. A carbon-hydrogen bond is 1 angstrom. So that is what you will see. So the error is so less in this protein structure. So AlphaFold has done really well. That is represented in all the superpositions that you see here. Again, blue and green represent, one is AlphaFold, one is experiment. And you will see not only at the level of the skeleton of the protein, but also at the functional sites where, oxygen is held here by amino acid residues. You will see all of those kind of things where it's superposing really beautifully out there. Whether in a small protein or in a big protein out here, AlphaFold has done very well. So that is what has demonstrated, and AlphaFold 1 to 2, it has evolved so much more. So now, in a way, we can say that the challenge of structure prediction has been solved, okay? Because you can estimate, you don't need to do X-ray crystallography for these kind of proteins anymore, because for new proteins, it may still be necessary, but for proteins for which there is a very strong chance of predicting a good structure, it may not be necessary. You can use it for other purposes, right? So that's where AlphaFold has hugely contributed by Demis Hassabis and John Jumper. Now I just want to compare quickly AlphaFold versus Rosetta. Rosetta was another modeling pipeline developed by David Baker and colleagues, which started as a small, laboratory exercise, but then later reached the community in such a nice way because they opened it up for as folding at home and exercises of that kind, like a community modeling exercise and so on. It differed, and today we have Rosetta Fold, where they have also encompassed artificial intelligence into their algorithms. While AlphaFold was data-driven, Rosetta was physics and knowledge-based, like the physical principles that I told you. So for different steps, they use different methods. And this here, it used Monte Carlo sampling for looking at what sort of a shape would be best suited for a sequence, whereas here it was AI-based understanding and so on. I won't go into all the details, it'll take too much time. But then David Baker and colleagues used Rosetta platform to ask a different question, right? What we've been talking about so far is, what is the structure for a sequence? Given a structure, what is the, given a sequence, what is the structure? Now you can ask a different question. Given a structure, what are all the sequences that are possible to fit in to that? Is it one? Is it many? And so on. So it's the inverse of that question. So which helps you go into another completely different domain called protein design. You can design new proteins, which is what Baker has shown so beautifully. In fact, as he says, we've entered an era where we can not only understand biological systems, but also create new ones, right? And that is why design is so important. So how does design work, right? Now design, you start with a two-dimensional blueprint. You have some imagination, some fancy of what you want, right? You make a two-dimensional blueprint of that. And you assemble the designed backbone from fragments of known structures. Take a bit from this protein, from that protein, so on, put them all together. And optimize it using the principles of physical principles underlying protein structure formation. You use them for design as well. And make changes as you require and finding conformations where with the lowest energy kind of state. So what do you get from this? You get a sequence independent, a novel protein structure. Sequence independent in the sense, we are not using the principles of similar sequences to similar structures. You're not saying you've got a structure. What are the sequences that are compatible with that, right? Overriding or bypassing the principles of evolution that we have learned. One can argue that, would evolution not have sampled these? Answer, maybe yes. If we have had million years or millions of years, maybe this protein will come up on its own naturally through the process of evolution. But then we definitely don't have that kind of time. At least in our lifetimes, we will not be able to understand anything about what evolution does if we have to wait for the natural time scales. So here you are trying to bypass or skip that step and then see if you can forcibly make a protein that is stable using principles of protein design. That is what they actually show. So they design proteins. But then there is a question that comes to anybody's mind. Yes, you can design something, but how does it fare in real life? Right? They showed that also beautifully. I will show you the protein they designed first, which was cited in the Nobel Prize. So what Baker did was to take that protein called TOP7 and made a synthetic gene out of that. One can actually synthesize a gene. In fact, we've also bought genes like that. You synthesize a peptide and you buy genes. You can synthesize a polypeptide like that. There are companies which sell synthetic genes. Then you insert it into a bacteria like E. coli, where you can grow in the laboratory quite easily using molecular biology techniques. You grow the cells, you purify the protein and determine its crystal structure. And in this case, not only could the designed protein fold, but it was exactly the same as the prediction, which was so beautiful. I'll show you that in a moment. This was the two-dimensional blueprint that they came up with. The protein was called top7. So 93 amino acid residue designed protein. These are individual amino acid letters drawn out as a 2D map. And it shows which are helices, which are beta sheets, which are secondary structures of this protein. And this is what they actually got. Blue is the prediction, red is the actual crystal structure, and it overlays. This is a stereo image. So if you are trained to see stereo images, the left hand and the right hand, if you stare at it for a few minutes, a few seconds, you will be able to see a three-dimensional diagram quite beautifully out here on top. And this is showing the portion of the same thing in a different visualization, different representation, where you very nicely see a helix and a beta hairpin out here coming together along with the amino acid residues. It's a representative region of the same is also shown out here. So the design and the X-ray salt structure were the same, which showed many things, which showed that one can actually design a stable protein fold. One can make a gene for that, and one can actually express it and then purify and solve the structure, and that will be the same. Okay. At this point of time, this protein did not have a function. Okay. It was only proving the principles of protein design. But very quickly, because the Baker's group had mastered this process, now they could scale up to a variety of different designs, variety of different proteins, and then they made many proteins. And in fact, I think many of them have commercial prospects, in the sense they have such huge applications, biotechnology applications. So they, I think Baker and his team have founded many companies, many startups, each one looking at one particular protein and its application, which will come into the market quite quickly, I'm sure, right? So some examples are shown here. This is a new nanomaterials. They're up to 120 proteins spontaneously linked together. You can make a cage, you can make a beautiful kind of a design which can have many applications, right? This particular one is a design protein which can bind to an opioid. It can detect, for example, fentanyl in the environment. And this one is again a nanoparticle which imitates influenza virus that can be used as a vaccine. And out here is a protein that can function as a molecular rotor, which rotates quite easily, which can have many molecules, which can be a synthetic molecular machine. And this one is a protein which is shaped in a certain way, which can change its shape based on external influences, which means it can serve as a beautiful sensor of something, right? So these are all the various proteins, shapes that can come out with. And both AlphaFold based predictions as well as Rosetta based designs have many applications out there. Like I said, Baker has co-founded several biotech companies. Many names are given in Wikipedia. And AlphaFold has been used in advancing drug discovery. Some examples are shown here, like these little ones are the drug molecules in yellow that you see here. And what you see as ribbons out there are portions of the protein molecule, which are good drug targets for whether it's for cancer or whether it is as for obesity. These are protein molecules which are good drug targets, and drugs have been designed, and I think various stages of clinical trials, which may enter the market quite quickly and so on. So the outlook for the future is fantastic. You will get new insights into biology, structure-informed biology. You'll get accelerated drug discovery. The way we think of drugs itself will be very different. In fact, I've been teaching a course on drug discovery for the past 25 years. We'll have to rethink now how discovery and design should happen, and how advances will go to the next level, next dimension, and so on. New drugs, advanced delivery vehicles where it is delivered only to cells that are required, and smart therapeutics which will adjust its dosage and deliver as and when it is necessary, right? All of these are possibilities today. And vaccines, also next-gen materials to put into delivery vehicles or for a variety of other purposes. You will have all of these things which are possible. What the lesson for us is, we have to retrain ourselves with the advances that are happening, and then see how to make best use of it and where to go from here. That is what we should, the younger generation scientists, as aspiring scientists, should be thinking of. And I'm sure we will see leaps and bounds of success in the years to come. Thank you for your attention.
That you taught. From basics, you took us to how deep neural network helps in unraveling the protein structure predictions and use of evolutionary information, then AlphaFold 2 architecture, then the training module, then critical assessment of protein structure prediction. And all these helped us to understand how biochemistry and AI and ML could help us in development of new drugs and new vaccines, and smart therapeutics, etc. Really, it was a wonderful exposition of the Nobel Prize winning research by you, madam. On behalf of Karnataka Science and Technology Academy, we are very thankful for your excellent talk. Now we have with us Professor H.S. Savitri madam and Professor Kushal Das sir. Now over to you, madam, for your comments, and also to sir for your comments. Madam Savitri madam. Sir Kushal Das sir, over to you.
Very good. Very good afternoon, sir and madam. It was an amazing talk, wonderful, and so beautifully you explained some of the complex mechanism which actually person like me who is a medical science background being a medical person, difficult to understand, but it is so easy way you have explained this part. So lot of thanks to you, madam, first.
Thank you very much.
And I just one thing that amazed to know one thing from you, that this use of the machine learning technique and artificial intelligence to predict and make it to a real shape, a design of a protein and development, what will be the adverse effect or any other safety is need to be concerned? Otherwise, a challenge may be faced in future to develop certain type of specific antigenic or antibody responsive protein in advance to prepare to make a biological warfare.
Absolutely. You touched upon a very important topic. Any technology will have great benefits, but it can also have dangers. It must be used wisely, which means regulation is necessary. Some sort of regulation policy must come in to see that these are not used for bioweapons or any bad things of any kind, for example. So that I think only legislation and policy will be able to control, scientifically informed policy. That is, I think, absolutely essential for that.
Thank you, madam. This is actually I was concerned about this one. It is wonderful for developing specifically neurological aspect by which you have explained also that how the neuronal network artificially it can be controlled and monitored. And being a medical person and a neurophysiologist myself, I find that it is very good for tomorrow's many of the disease specifically to target specific protein and to modulate in that way. But only I am worried about that one which just now I shared with all of you. Thank you, madam, for wonderful talk and enlighten us with your beautiful experience of your own and sharing some of your moments and all with the Nobel laureates themselves personally. That's a great addition to us as a gift. Thank you, ma'am.
Thank you.
Thank you, sir. Now, Professor Savitri madam is there with us. She was a member of KSTA, and during the previous committee, madam was also a member. We started this Nobel Lecture Series, which has been very useful and many people have appreciated. Your talk was superb. Thank you very much. Now, over to Madam Savitri madam.
Excellent talk, Nagasuma. It was very clear and taking from the beginning to the current research scenario. You could speak so authoritatively because you have been through the entire process. You have been working and moving with the Nobel Prize winning work as well. So it was really excellent and nicely presented. And now the question comes whether, then I saw this in some of the recent papers that they are saying you look at a cell and then use high resolution microscopy and so on and so forth, so that you could tag every protein in the cell and say where it is going, with what it is interacting, how it is working and all that. So it's really AI and ML, along with the experimental work that is already accumulated so much. One day we may be able to look at a cell and see how the communication is happening with hundreds and thousands of proteins and other molecules, and how the cell is able to perform such an intricate function. So in that sense, I think, as you said, the future is very bright and there are so many things that one can work on. Thank you very much. It was a wonderful talk.
Thank you so much, Professor Savitri. I must also say, Professor Savitri has been like a mentor and senior colleague to many of us, and it's been wonderful.
No, that is all not so much. It is all your work. You have done so well. But anyway, I'm so proud of you.
Thank you.
I'm also very thankful to Dr. M.R.N. Murthy sir, who suggested your name when we came up with the Nobel laureate lecture on chemistry. I'm very thankful to M.R.N. Murthy sir also. He only suggested Nagasuma madam for this talk. Tomorrow he'll be again giving a talk on different aspect of this Nobel Prize. And are there any questions, Umesh?
There are only 2 questions. I will take them now.
Please then.
Sequence or a single chemical compound?
A sequence by itself. Okay, one peptide chain, we call it as one protein, one sequence. It can have multiple conformations, in the sense multiple shapes. It's like taking a bead of a chain of beads, and can you not fold a chain of beads in multiple ways? And that is what we are saying is by multiple conformations. But nature will see to that only the least energy or the most stable shape is what will actually occur. And that has been programmed through evolution, through the choice of the amino acid residues itself in the peptide chain, so that some function is there. We don't have any wasteful protein. So any shape that is there will have a specific function. And that is how evolution would have selected that.
Thank you very much, ma'am. There is one more question from Dr. Vijay TS. It's related to the gene expression by AI models is mimicked by the past data. How to exactly predict the nature's intention, which codes and sequences protein in a particular way?
Okay, let me understand the question. The gene sequence, and it's the sequence that is read out as a structure. Expression is a very different thing. Now, expression is how many copies of protein molecules will be made from this gene once it's expressed. That is gene expression. AlphaFold does not look at it, but there are other AI models, there are other data analysis things that we do in the lab and many others do for understanding that. So that maybe I should answer that question in a separate occasion. How to exactly predict the nature's intention, which codes and sequences protein in a particular way. So the bottom line here, okay, so the sequence, any change in sequence should not interfere with that function. It should facilitate that. So if it is an RNA polymerase, it needs to remain as an RNA polymerase. So that is how it knows what to do.
Thank you very much, ma'am. One more question from Vijay TS is, when we mutate the virus for discovering new vaccines, does it pose a danger to public health by forcing artificial mutation instead of natural mutation?
Yeah, you can mutate the virus. In fact, people don't take the whole virus and mutate. That's the conventional way of discovering viruses also, or by attenuating them in some sense, right? Even bacteria, you subculture bacteria, for example, or you attenuate viruses to see that they are not virulent. Yet some mutations have happened, and where they can pass on some protection in the form of immune response and so on. So this is not a new thing. This is how it actually happens. Like we answered before, any technology can have a danger if it is used wrongly. But if it is used correctly, there is no danger to public health. In fact, our Covaxin will be that kind of a vaccine where some mutations will be there. Whereas Covishield and other things which are subunit based vaccines will have only one portion of that thing. So mutations are the way to go forward. That is there. Right? You are forcing, I think, to add to the answer, there are things called gene drives that people do, like for a mosquito population or whatever, they put in some mosquitoes where they are not virulent. I mean, they're not carrying any pathogens, but they mix with the mosquito population to see that their numbers grow, and the ones which may be carrying something or which have the ability to carry some viruses or other pathogens are reduced in number. So these kind of things are done at different levels.
Thank you very much, ma'am. Madam, few of the research scholars are requesting your contact details if they can communicate with you if they come across any doubts in future. So, shall we share your email ID, ma'am?
Email address you can share, yes, please.
Okay, ma'am. Thank you very much. There is one more last question, ma'am. What are the future perspectives in the field of proteomics for early career researchers in biotechnology?
Yeah, there are plenty of prospects of that. Proteomics is now nothing but looking at all the proteins that are there in a given cell. So that is proteomics. So as Professor Savitri was saying, we may be able to look at spatial proteomics in a cell, like what are all the proteins, how is communication happening, how is signal being passed on from one protein to the other, and how enzymes work in coordination with each other and so on. There's a lot of scope for that. There's also a lot of scope for applications and jobs, careers based on that, biotechnology careers. So I think there is plenty of scope.
Thank you very much, ma'am. That's it.
Okay.
Madam, thank you once again. Thank you very much for your wonderful presentation on behalf of KSTA and all those who have joined the webinar on Cisco WebEx and YouTube. Once again, thank you very much. And I also this webinar, your presence has really inspired us. Thank you very much, sir and madam. And I'm also thankful to Dr. Senior colleagues, Dr. Dhulappa, Dr. S.B.N. Murthy, Dr. Sanat Kumar, Dr. Qureshi, and Professor Shivanand for joining the webinar. And also I am thankful to Miss Garima, Vijay, and Ali Javed for posing such good questions to understand the subject better. I urge youngsters to ask experts many relevant questions and clear their doubts when experts are here. After that, even if you want to try to connect them, it will be difficult because most of them will be busy with their own work and with their own research. And tomorrow, again, Nobel Lecture Series on chemistry. One more talk will be delivered by Professor Namaran Murthy sir, former Dean IISc. So all of you join by 11:30 and we will be having the talk by an expert.
Thank you, everyone.